And if you could just press stop, it would be awesome.
Okay, good morning. Let's get started. I wanted to thank those of you that were able to actually make it over to the annual meeting of the Center for Membrane Protein Research. A lot of it is pretty complicated, and some of that we'll actually talk about today. In fact, two of the main topics that they talked about were pumps and actually secondary active transporters. That was Dr. Ernest Wright, and then several ion channel people yesterday. So we're really not to the place where we can understand everything that they were talking about, nor will we ever be, but unless you go and work in their lab, which is quite possible. So if you're interested in working with some of these people, what you need to do is go to the Cell Physiology and Molecular Biophysics website, open up the box for the faculty members, and then figure out what go to that particular faculty member and get their contact information and send them an email. But the last thing you want to do is you don't want to send a blanket email, please take me. You want to send individual emails to those people that you're interested in talking to, because that's how you start a conversation. But don't go there unless you've read some of their stuff so that you have sort of a working knowledge of what they do. They can't possibly expect you to know everything, but you need to come in with not completely naive either, okay? So I hope some of you will actually take that opportunity and do that. And if you want to talk to me about how to approach that, please let me know. I've set a lot of undergraduate researchers on their research paths before, so I know how it works, okay? We needed to finish one more pump, which is actually not a pump, but it looks like and smells like a pump. It's actually an ion channel. And then we're going to get into secondary active. So we need to complete the primary active stuff. And our poster child for the ABC transporters is the MDR1 pump. Also in that family, and the reason I bring it up, it's very medically related, is the cystic fibrosis transmembrane conductance regulator. And nobody ever says that. They always say CFTR, okay? And that's why. And so we need to look at that pump. It's in a pump family, but it actually is an ion channel. And here's the structure that tells you that it's in the ABC transporter family. It's got two different membrane domains. Each one has 6 alpha helices, so that's a total of 12. And so therefore you know that if the N-terminus is cytoplasmic, even number of alpha helices, where's the C-terminus? It's going to be in the cytoplasm, okay? So you go up and down, up and down, up and down 6 times, and you come back out, and then there's this little bump here, and this bump is the first nucleotide binding domain. And in people that have cystic fibrosis, it's usually 2/3 of the cases it's amino acid 508, which is a phenylalanine. It's one amino acid deletion. And the reason that person gets cystic fibrosis is not because the pump won't work, it just never gets to the plasma membrane. It gets inserted into the ER, as you'll see in the next segment, when we start talking about protein trafficking. It gets going on the process, but it misfolds, and misfolded proteins are ejected from the ER, and they never make it to the plasma membrane. Now, that's at body temperature. If you could chill this person down to about, I think, 10 degrees, 15 degrees centigrade, he would die, of course, but the pump would work. Because at that temperature, it folds correctly and is trafficked to the plasma membrane and operates as a chloride channel. Okay? But this is supposed to be an ABC ATPase, right? It's got 2 nucleotide binding domains. It binds ATP, but why is it a chloride channel and not a pump? Okay, that's the question we have to ask. But let's step back and look at the physiology to give us some sort of context about what the function is of this membrane protein. So this is an airway, okay, in a normal person. And so here's the epithelium, and this is a fluid transporting epithelium. And the reason it transports fluid is, so what's going to happen is sodium leaks through this paracellular pathway. It actually doesn't go through the cells. Some epithelia are leaky. That means the junctions right there and right there holding the epithelia together is strong, but it's leaky to certain ions. So to get sodium to move, what you have to do is you need to transport chloride into the lumen. And that's what the CFR does. But chloride can only go through that CFTR protein if it's going down its electrochemical gradient. Okay? And so there's some pumping going on. There's actually a bicarbonate chloride exchange process that's not shown here. That builds up a gradient of chloride, and then so chloride's going to diffuse through this ion channel into the airway. That creates a negative potential that's going to suck sodium this way through the paracellular pathway. So now you have sodium and chloride, and that's going to drag water osmotically through the cell into the lumen. What does that do for you? It washes out all this buildup of mucus. The mucus is there to keep the bacteria away, but you have to continually clear out that mucus, and to do that you need to sweep it away with a lot of fluid. So a person with cystic fibrosis, the chloride channel does not work because it's not at the plasma membrane. It's been destroyed. Okay? And so there's no pathway for chloride, so there's no way for sodium to get across. And so what does that mean for water flow? It's blocked, isn't it? Because you can't create the sodium chloride gradient to pull the water across. What happens is that mucus builds up, the bacteria stick in there, and they infect the lining of the airway, and so that person gets an inflammation, it closes off, and they have really bad trouble breathing. In fact, one of the treatments is actually hang them upside down to help get rid of the mucus out of their lungs. And it's not only a problem here, because unfortunately the CFTR is also located in your sweat glands. So they have problems with ion. They lose lots of ions because they can't use the chloride channel to bring back the sodium. So their sweat is very salty. Okay? And the other thing is in gastrointestinal system, you secrete lots of fluids, and in some cases in the intestine, you actually use this chloride channel to move food up and down the GI tract. So they have problems with digestion, breathing, body temperature regulation. So there's lots of different problems. Okay. It's all because of that little guy, the chloride channel. So how does it operate as a channel? What's going to happen, and we're not going to, I'm not going to detail all the regulation, because what's going to happen is some hormone, the neurotransmitter, is going to talk to the cell and say, you need to open that channel. Okay? And the result of that hormone or neurotransmitter binding to that cell is going to create a signal inside the cell called cyclic AMP. That's, you probably have heard of second messenger. Cyclic AMP is a classic second messenger. And so it's going to instruct the cell what to do next to move the chloride. And what cyclic AMP does is it turns on this enzyme called protein kinase A. So that tells you what it does, right? It's a protein kinase, so it's going to phosphorylate some protein. And the protein that PKA, protein kinase A, phosphorylates is actually the CFTR. It phosphorylates it in a regulatory domain, and what was happening before cyclic AMP turned on this kinase is that regulatory domain was preventing the channel from opening. It's stuck there, right between the two nucleotide binding domains, this big old bulge. Most ABC transporters do not have that regulatory domain. Okay? So you have to do two things to open the chloride channel. First of all, you have to phosphorylate the regulatory domain, and it causes it to bristle with negative charges. It falls away from the nucleotide binding domain, and that allows the nucleotide binding domain to bind ATP. And so what this is an ATP-gated channel. The ATP is actually split, but there's some argument about that. But the ATP binding to the nucleotide binding domain is what actually opens the channel. And when you open the channel, you're not actually ejecting something, you're just creating an aqueous pore for chloride to move. And chloride will only move, for all channels, this is a good generalization, the solute can only move down its electrochemical gradient. So you have to have built up enough chloride inside the cell for it to diffuse down its electrochemical gradient. Okay. And now the electrical gradient is favorable, right? Because what's the voltage on the inside of the cell? Negative 60 is the Joe Blow membrane potential. Okay, what's the potential outside the cell? Zero, right? Okay, so it's positive outside the cell relative to that chloride ion, so that positive potential is going to help move the chloride down its electrical gradient, and then you also have a chemical gradient of chloride that builds up inside the cytoplasm, so it's really going to zip out. So you have to have two things to move the chloride. You have to have a favorable energy gradient, and you have to have a pathway. So you have to, until this is open, you cannot move chloride. So you must have a pathway to move the chloride across the membrane. That pathway is the protein CFTR. Okay? Does that make sense? All right, do I have any questions about it? What I want you to ignore is, so hormones do something, and we're going to talk about that in about 3 weeks. Hormones do something to a receptor that causes a little pulse of this chemical to form, and that chemical is going to activate a kinase. Not by phosphorylating it, but just by binding to it. Okay? And that kinase starts the ball rolling, phosphorylating, and opening the regulatory domain, moving it out of the way, ATP binds, and then the gate opens. Okay? So this is, we're going to talk about different types of ion channels. Where would we put that in the category? It's a ligand-gated channel. That's where we would put it. And it is not a pump. Not a pump. Okay, any questions about the pumps? So these are all primary active transport mechanisms except this weird one that we just got through talking about, which is an ion channel. So that's always passive, okay? So let's move to the topic of secondary active, and to do that we need to get out of this outline and go to this one. So secondary active has lots of different names, and so one of those is cotransport. You have to have at least two different solutes moving to be secondary active. Solute number one, and this is the simplest case, solute number one is going to provide the energy by having a favorable electrochemical gradient. It's going to move down its electrochemical gradient. That's going to force the protein to fold and capture a second solute, which is going to move up its electrochemical gradient. And the reason it can do that without any ATP is that it's using the energy from the other solute's electrochemical gradient. Okay? But generally what happens is that first gradient that's favorable for the first ion has been established by a pump. So ATP is indirectly involved, and so that's why we call this secondary active, because there's no ATP hydrolysis by this protein. Okay. So our poster child, and we will have two, I've already talked about one, which was the sodium-calcium exchanger, but let's do this. So this is the sodium glucose cotransporter. Dr. Ernie Wright from UCLA that I used to work next to for about 5 years, they cloned this protein. Okay, so we know a lot about it because of Dr. Wright's lab, and so let's take a look at it. There's something wrong though actually with this statement. This is what most textbooks say about it, and if you went to his lecture, you could probably tell me what's wrong. So all cells have one or more glutes, okay? In contrast, only two tissues can actively take up glucose, the small intestine and the kidney tubules. Is that still true? Did anybody go? A lot of you had class. What's wrong with that? Do you remember?
Muscle cells. The PET scan showed muscle cells.
Yeah, there's a number of things wrong with this. Number one, there's not just SGLT1, there's 12 of them, 12 different isoforms. They're scattered all in heart muscle, in the brain, in the blood-brain barrier, in places you wouldn't even realize. Okay, so it's naive to think that there's only 2 places where you can actively transport glucose. In many cases, we don't know what the function is for those pumps, but we do know in a couple of situations that become very clinically important. So I'll describe that to you after we've talked about the basic physiology. Let's talk about the first one that was discovered, okay? Actually, it was Dr. Wright was the PI in charge, but his right-hand man was a postdoc like I was. We were good friends. Matthias Heidegger, he's Swiss. He was working at UCLA, and we used to compete for who could stay the latest and work on proteins, okay? He usually won, and the reason he did, he had an unfair advantage. His wife worked in the lab with him. I had to go home to see my wife, so I had to leave early. So that's my excuse anyway. So what Matthias did is he actually size fractionated all the RNA in a cell, all the messenger RNA, into different sizes. And so he would take this size of RNA and he would inject it into a Xenopus oocyte. And the nice thing about a frog egg is whatever RNA you give it, it'll make that protein for you and display it on the plasma membrane. And so the assay was inject the RNA and see if you've got glucose transport. And he just did this over and over and over until he found the right fraction. Okay, then once he got that, he broke them down into smaller and smaller fractions until he had but one RNA species, and that was the SGLT glucose cotransporter. Okay, so this stands for sodium glucose cotransporter, and since it was the first one, it's called SGLT1. Right? All right, what does it look like? We'll come back and do those. You need to go to lecture 10. All right, just to remind you what's going on here, the SGLT1 is a cotransport, but that means just more than 2 different solutes or more than one solutes being transported in some direction. All right, but you have different types of this, right? So this is the coupled cotransport, is just a short term for coupled transport. The SGLT1 is a symport because both solutes, they're not substrates now, they're solutes, are moving in the same direction. So what's moving, it actually is 2 sodiums going down its electrochemical gradient and 1 glucose molecule going against its electrochemical gradient. Okay? So the sodium gradient is powering the movement of the glucose into the cell. So we call that a symporter. Okay? If the 2 different solutes were moving in opposite directions, like they do for the sodium-calcium exchanger, we call that an antiport. So we're going to focus on the symport first. Okay. Now, this is an okay figure because it sort of tells what's supposed to be happening. What happens is the sodium loads onto the transport protein when it's in the T1 configuration. It's facing outside towards the gut lumen. Let's use the intestine as our... So this cell, these cells that have SGLT1 in the intestine face outside, face the gut lumen. So they're there to pick up what? Glucose that you've eaten, right? All right, so the first thing that loads on are 2 sodium ions, and then that actually opens up a gate that allows glucose to come on. But it's not 2 glucose, so that's what's wrong with this picture. It's 1 glucose. 2 sodiums, 1 glucose. Okay? And what happens is when everybody is loaded onto the pump, or I'm sorry, the cotransporter, it then refolds, okay, so that the ion and sugar binding sites face the cytoplasm. Okay? And it turns out that the sodium comes off first, okay, and then the glucose comes off. It's lost some of its affinity for both of those, okay, and so the sodium diffuses into the cell, the glucose diffuses into the cell. The empty pump then refolds by itself to face the outside to pick up the next round. And that refolding of the unloaded carrier is, again, the rate limiting step. Without any solutes, it's really slow to refold. Okay. So this happens about, maybe 100 to 1,000 times per second. So it's faster, it's much faster than a pump, but it's slower than an ion channel. So these co-transport mechanisms are sort of intermediate between the pumps, which is the absolute slowest, because you've got to break ATP, and you got to phosphorylate it, and you got to flip it, and all that other stuff has to happen. This is a little bit faster. Okay? So you can pull in lots of glucose pretty fast. So here's a more detailed look at this. So the unloaded transporter is facing outside. 2 sodiums bind. The stoichiometry is correct here. That allows 1 glucose to bind. And now it's ready to reorient from the T1 to the T2, which means it's facing the inside of the cell. So this is the cytoplasm. And the sodium is going to leave, and then the glucose leaves. What happens to the sodium? It's expelled. By what? What expels it? Sodium, potassium pump, right? So that's where you're creating the sodium gradient, is because the sodium pump is continuously getting rid of the sodium to lower the concentration of sodium so that you can bring in more sodium and more glucose. Right? And then the glucose is released, and then it's shipped into the bloodstream for the use for the rest of the body. Okay? So that's how this works. And so when you lose both solutes, then this thing refolds back to the T1 very slowly. That's the rate limiting step. Now, do we have a sort of drug tool that we can use to inhibit this? Yes, it's called fluorescein. And it's spelled, it's an ugly looking word. It binds to SGLT1 with very high specificity and blocks the transport of just SGLT1. Okay. All right, so now let's do something kind of useful with this, because just having an SGLT1 available is not too exciting unless you can actually move glucose into your body from the gut lumen. So how does that actually work? Well, then you need to get in, you need to put the SGLT1 in the context of, we've already talked about the sodium calcium antiporter, so I'm not going to talk about, that's our poster child for the antiporter that we already talked about. And so we've got an epithelial transport. So the gut lumen, or the gut absorptive cell, is structurally polarized. I'm not talking about electrically polarized. Obviously it's electrically polarized. But what that means is it's got a different set of proteins on the apical side than it does on the basolateral side. So what defines the apical from the basolateral? The tight junction does. Okay? So the tight junction are proteins that are holding this epithelia together. And it creates a diffusion barrier, so the glucose can't come across here, and the sodium can't come across here. It has to go through the cells. And that gives the intestine control over what solutes it wants, and which solutes it leaves behind and are eliminated. Okay? So we want glucose, and we want sodium. So what we're going to do is couple those two by, this is SGLT1 at the apical membrane. So it's 2 sodiums, 1 glucose. This thing flips around, dumps the glucose in. The pump is actually located on the basolateral side. So the sodium pump is not here, and there's a really key reason for that. So we're going to put it here, okay? This is the basal, so that's the tight junction. Everything above it is apical. Everything below it is basolateral. So this is basal, and this is lateral. So we combine those two words to show this perimeter of the cell is the basolateral membrane. And it's held there intact by the tight junctions. So the sodium pump cannot diffuse all the way into the apical membrane, and SGLT1 can't diffuse into the basolateral membrane. So then the sodium, 3 sodiums are expelled, potassium is brought in. That creates or contributes to a negative membrane potential. That's important because that negative membrane potential is going to help suck glucose in. Okay? And because it's expelling glucose, it's going to keep, expelling sodium, it's going to keep sodium nice and low. So the 2 driving forces needed to move sodium in are present. Low sodium and negative membrane potential. Okay? And then glucose gets a free ride. It's actually can be concentrated several hundred-fold inside your cell. Okay, so we're going to build up a high concentration of glucose. How is it going to get into your bloodstream? Your bloodstream's out here. Okay? Well, we need another transporter, and that transporter is an old friend of ours. It's GLUT2. GLUT2 is on the basal-lateral membrane. Why do we choose GLUT2? We don't need a pump. We don't need another active transport mechanism because your blood glucose is what, about, I think it's 5 millimolar, a normal blood glucose level, okay? And this is much higher than that. So glucose is going to go down. It's a chemical gradient. There's no electrical gradient. Glucose is polar, but it's not charged. So all we need to do is provide a pathway to get across the lipid bilayer, and that pathway is GLUT2. Now, what do you remember about GLUT2, if anything? Remember, we put different isoforms of GLUT based on what? Based on? Yeah, that's true. And so we can put a really sloppy GLUT2 member here because it encounters really high concentrations of glucose. Not because it's experiencing them out here in the lumen, but because the cell is concentrating glucose inside the cell. So this thing doesn't have to be doesn't have to have a strong affinity for glucose. It's going to go diffuse down its chemical potential into the blood, as long as you have normal glucose. What if you have a diabetic? What is that going to do to your intestinal glucose absorption? Absolutely. So if your blood glucose rises, that's going to slow it's actually good for that person. It slows the rate of glucose entry because blood glucose is high. This could get, it gets really high, so it never stops the glucose flow, but it slows it down in an uncontrolled diabetic. Okay? So that's not good, right? What happens is if you fail to transport all the glucose and sodium into your blood? Let's say you leave behind, let's say for some reason, so a diabetic may not be able to pull all of the glucose into the lumen before the food moves all the way down the GI tract. What happens? If it enters the colon, this is all in the small intestine. Okay? That's where glucose transport is highest. You get diarrhea, okay? Because osmotically, those nutrients will pull water back into the colon, and the lumen will fill with water, and you get diarrhea. So and people can die from dehydration, right? And that's what kills people with cholera is the gut just cannot absorb fluid; it secretes it, and then those people die of dehydration. Okay? So you do not want glucose going into the colon. That's lethal. Okay. All right. So. SGLT, I'm sorry, this is GLUT2. The pump operates. The SGLT1 is pulling in the glucose actively. Now, another reason you want to put GLUT2 here is it doesn't only transport glucose, it transports galactose as well. So it's not because it's got a sloppy affinity, it doesn't need to have a high affinity. Why? Because you're going to stack up concentrations of glucose very high. So you don't need a high affinity, but it also means it can carry more than one passenger. So it will actually absorb not only glucose, but galactose, and it will actually transport fructose. So there's 3 different sugars that SGLT2 will handle. I'm sorry, GLUT2 will handle. All right, so where does galactose, how does it get into your cell? The SGLT1 will transport galactose just as equally well as glucose. So it is a glucose-galactose active transport mechanism. So when do you get galactose? What do you eat to get galactose? It's not what you eat, it's what you drink. Milk. Okay. Lactose is split by an enzyme, okay, on the apical membrane. And let's draw that picture because we need to. So things don't, it's not this simple. When you eat something, it doesn't just immediately go into your bloodstream. There's some basic physiology that we need to consider. So, oh, yeah. Good. Okay, so let's draw this way. And draw a little Bart Simpson. That tells you that which way is apical, because his spiky hair faces the gut lumen. There's a tight junction here, right? Okay. And more Bart Simpsons. Another Bart Simpson, ad infinitum in both directions. So the epithelium stretches across both sides. So this is the gut lumen up here. And this is a lousy pen. So okay. And this is the blood. Let's make it red then. Blood's here. Absolutely, yeah, thanks. This thing is so tricky. Okay. So now let's put all the players that we know about into the membrane. Let's put, so this is SGLT1. There, we know this is the sodium pump. There, we know that it's different colors in here. This is GLUT2. There, okay. Now, what's happening is, so you take lactose, and there's an enzyme, a digestive enzyme, that is, it's a single pass membrane protein. Okay, it's actually inserted in the apical membrane. Its active site is facing the gut lumen, and so lactose binds to that, and it gets split into galactose plus glucose. So you get a twofer, right? Two different sugars. And the reason you're splitting it is you can't get those long polymers of glucose of sugars to go through your epithelium. So what the lumen's strategy is, you give me all these nutrients, I'm going to break down the polymers, the proteins and amino acids, the sugars into individual monomeric sugars from big starches, and then we'll transport the monomers in. It's much more easy to pull in. Okay? So lactose is split. Both of these two guys are going to go through here, not at the same time. You're going to get glucose binding, 2 sodiums, flip, goes in. Galactose binds, 2 sodiums bind, it goes in. Okay? Then they both, so you build up galactose and glucose at high levels in the cell, and they both independently go through GLUT2. Again, not at the same time, one by one. Down their chemical potential. Right? Now, what happens when you eat table sugar? Sucrose. So it's a similar thing. There's another enzyme, and it's called, strangely enough, sucrase. Okay? And so that's going to break down sucrose into fructose plus glucose. Okay. Now, you have a new player. Okay? Fructose is not transported by your SGLT1. It stays in the gut lumen. What will happen if you leave the fructose in your gut lumen? You get diarrhea and dehydration, so you need to transport it. But what we're going to do, think about your proteins that you've learned about, transport proteins you've learned about. Did we have one that transports fructose? It's a uniport. It's kind of strange because it's in the GLUT family, but it doesn't transport glucose. Its Km for glucose is infinite. It's huge. It means it can't move glucose. GLUT5 is the fructose transporter. So where are we going to put it? Which membrane? Apical membrane, right? So we're going to put our little friend GLUT5 up here. And that will take fructose. It's going to have to dodge around all the green things and then come in. And so now you have fructose inside the cell. How do you get rid of it? That's where you use your old sloppy friend, GLUT2. It will be transported in blood through GLUT2, because GLUT2 is very sloppy. That's entity. It will accept fructose, lactose, and glucose. So now when you eat these complex sugars, these are actually pretty simple sugars, right? These are disaccharides, but generally they're broken down into the monomeric glucose, fructose, and lactose. So now you have a method of getting that across. And actually, you're pulling sodium and glucose, so osmotically what's going to happen? You're going to pull water through, and so that also returns back to your body. Because when you digest stuff, you secrete more water than you drink during the day. And if you just left it there, you would die of dehydration. So it's important, with all that secretion going on, to move food, you need to pull it back in so you don't become dehydrated. And your body takes care of that without you taking it. Okay. I think that's all I wanted to say there. All right. So before we leave this little topic, let's talk about what these other isoforms may contribute clinically to our understanding of certain diseases. Okay. So let's talk about, so this was the first one that they cloned, and once you have the structure, you can get really specific about looking for genes that might encode these proteins. So pretty quickly after that, the Wright lab, Ernest Wright lab, also isolated SGLT2. And what they found is SGLT2 is mainly expressed in the kidney. And if you went to his talk, you would recall that he started his sort of his trek in science, thinking about how the renal nephron, the absorptive unit of the kidney, actually moves glucose out of the urine back into the bloodstream. Okay? So let's think about that. So now we're going to go to the kidney and use the same transport. We don't have to learn anything new about the transport cycle and all that. It's moving 2 sodiums and 1 glucose. But what's happening is, so you have your bloodstream breaks up into capillaries, and then it forms this little ball called the glomerulus. Okay, so blood's coming in, it forms a capillary ball called the glomerulus. That's Latin for little ball. So, yeah, how inventive. Okay, so the blood flows through there, and all the good stuff that's small is actually moved into this renal tubule, this little green thing. It's actually called, we don't need to know all of the components, but that's the Bowman's capsule. Okay, so everything small goes in here, and all the proteins stay in the blood. You don't want proteins going into your nephron. If that happens, it means you have kidney disease. Okay, that's the first sign that you look for present. If you have proteins in here, that's bad, even with small proteins. So what's going to go through there is all your amino acids, your glucose and nucleotides and everything. And so if you just, so this is going to form urine. If you just peed it out, you lose all those nutrients. But the body is smarter than that. So what it does is it takes a little tubule that's called the proximal convoluted tubule. It's convoluted because it increases the surface area, convoluted tubule. And that is where the glucose is pulled back out. And so right up front, it's going to take the glucose back and return it into the bloodstream. All right. The thing that does that, so let's put a little Bart Simpson. So this is now not the gut epithelium, but it's the kidney tubule epithelium. We've blown up this right here that way. So sitting in the apical membrane is not SGLT1, it's SGLT2. And you know the rest of the story. The glucose is going to come in, you have a sodium pump on the basolateral membrane, and the SGLT2 is going to send the glucose through GLUT2, GLUT2, out into the bloodstream when you get the goodies stuff. And this also happens for amino acids and different nucleotides. Right? So, now, the good thing about that is glucose should not appear in your urine if you are normal. Right? The capacity that this pump can handle is when your blood glucose, it has to actually go over 100 milligrams per deciliter to saturate that pump. Because there's lots of them. There's some SGLT1 here, but not very much. Okay? And so you have to have hyperglycemia to exceed that limit. And people that are poorly controlled diabetics will reach that limit at different times, right? Particularly after meals. Okay? And what happens is if your blood glucose goes above 180, that means the fluid coming in here has glucose at 180 or above the renal threshold for glucose. It's going to stay in the tubule, and it will go through the tubule, it will go into the urine, and osmotically it will dehydrate you because it'll pull fluid with it. Okay, so that's another danger independent of the glucose. Okay, so, but you don't worry, if you're normal, you don't need to worry about that because your blood glucose never goes above what? 100, 105 maybe? After a meal if you had a bad day? Okay, so, but this is a long way of saying, is there some sort of treatment that we could come up with that we could lower people's blood glucose if we fooled with the system? What if we gave that person fluorescein? What if we were stupid and we gave them a fluorescent pill? What would happen? Let's go back to the gut. This person's eating table sugar for some reason. Likes table sugar. Okay. Or drinking a cola or something, right? So glucose is in the GI lumen. You give them fluorescent. You want to get the fluorescent into the bloodstream to actually block this. Okay. So what's going to happen first? The person would die of diarrhea because the glucose would never make it into your body. It would block the glucose from entering, and that person would have contracted diarrhea and dehydrated. Okay? So you can't give fluorescent to do this. What you want to do is come up with a pharmaceutical that will block SGLT2 specifically. And why would you do that? Well, what's going to happen is if your blood glucose, you can actually titrate how many of the pumps, what fraction of the pumps you're going to block. So if we give a certain dose of the correct chemical, what it's going to do is it will actually lower the renal threshold down to 120. 120 is almost normal. So if we give that person a type 2, or this works for type 1 or type 2 diabetes, okay? You give them the right dose of a SGLT2 specific blocker, the set point is now at 120. If their blood glucose goes above 120, they pee it out. Their blood glucose is locked at 120. Cannot go higher. Okay? And most of type 2, the largest fraction of diabetes, 90% are type 2s. What other kind of problems do they have? What are the problems that you run? What is the outcome of having type 2 diabetes for a long-standing group? Pardon me? Yeah, lots of cardiovascular problems. And that's a whole different story that I talk about next semester. But they have heart disease. What kind of pill do you give a person that's undergoing hypertension? What's the you know what the first treatment, the simplest, best treatment that you start out with? If you have a patient that has hypertension, what do you use? You may not know. It's cheap. It's dirt cheap. Diuretics. Okay. And what that does is it lowers their blood volume, which volume returning to the heart is reduced. And Starling's law of the heart means that your blood pressure will go down. Okay, so this is a twofer. Not only do you lower the person's set point to 120, but you're giving them a diuretic as well, lowering the blood pressure at the same time. This is amazing. And so let's see if I have the drug. It's in, it should be pretty close to finishing clinical trials, but yeah, there it is. It has a horrible, I don't know why they choose these ugly names. DAPA with Flozin. The only thing that makes sense here is the Flozin that makes it flow. But that's they're working on lots of different drugs. Now, the nice thing about it is you can give this by mouth because it doesn't touch SGLT1. It's very specific for SGLT2. So it's an oral thing. You don't have to be injected. It goes right through the intestine. It hits the kidney, lowers blood glucose, and causes a mild diuresis. Okay? Unless that person gets uncontrolled and their blood glucose was really high, so you have to warn the patient what's going to happen. If it gets too high, you can really dehydrate. So that's a side effect that would have to be controlled. But based on, if you went to, there's only a few people in the back that went. The big problem, though, is all these other SGLTs that we're just now finding out about. SGLT2 is in the heart. What's it doing there? You're blocking it. What if its normal function is important? And your heart starts to palpate. So that's what they're looking for, is at what level can you give SGLT2 and not have any other side effects. So, okay, any questions about what's going on here? Okay. Let's take a break here. Hey.
So SGLT2, T1 and 2, they're pretty much essentially they have the same function. It's just SGLT,
Different places.
Different
physiological
Two is kidney and the heart, and then one is just generally located.
Pretty much in the intestine is where that is.
Okay. Cool.
Yeah.
Thank you.
Sure. Okay. You don't?
I don't know where it is. I thought it was
Okay, well, let's get started again. Couple of brief announcements. Where are you on your poster project? Okay, so what you've given me is a broad topic. So what I'm going to post today is a timeline of what you should be doing. Okay, and the next thing to do, so the quiz 3 is in 2 weeks, 2 weeks from last Tuesday, this past Tuesday. So it's sneaking up, and I'm not changing it. I'm leaving it there. The reason I'm going to do that is so that you have 2 grades before the drop date to make some sort of informed decision. Okay, so that's why I'm doing it. It actually reduces the amount of material, blah blah blah. So we're leaving quiz 2 exactly where it is on the syllabus. All right, that's one important thing. So you have a little bit of time though in the next week where you need to. Yes.
Sorry, what is the cutoff date for information on quiz 2?
On what now?
What's the information cutoff for quiz 2?
It'll be somewhere around next Tuesday. Yeah. We'll probably finish ion transport and that'll be phenomenal. So what you want to do is, so you're working on there's a lot of people that are interested in autoimmune diseases for some reason, that's fine. But you need to narrow what your focus is. Number one, which autoimmune disease? Type 1 diabetes, lupus, whatever. So you need to narrow it. And then within that disease, what specific thing are you going to look at? Okay, that needs to happen by next week, next Friday. And if you don't, you're going to get stuck, because then after that, you're going to be involved in studying for quiz 2. So you need to get with your group and figure that out. Okay. So what you need to do is read a couple of review papers. Everybody in your group you need to identify what it is that you're interested in, get some review papers, and everybody reads it, because the next step is everybody has to make an initial contribution in your group. Everybody has to come up with some approach on their own so that we don't have like 2 people in the group doing all the work. So that's called the initial contribution, and I'll post exactly, that's actually scored, okay, and what's involved with your initial contribution. Because I want everybody thinking about this approach, several different minds thinking, and you'll come up with the best one of those approaches, and that will improve your poster. And it also will get everybody involved in doing it, because the criticisms from people in the past, and I do listen to those, is that there are always a couple of people get stuck with doing everything. And that's not what you want to happen. Because you're going to rank each other at the end of the semester based on how much effort everybody put in. And what you don't want to be is left out on the side and having somebody else do all the work, and then you get a bad ranking, and then you lose group points. But you don't want that. So I'm sort of forcing the issue by this initial contribution. But you can't get to there until your group coalesces around a disease and an idea about what you're going to do. Are we going to inhibit this group of proteins, or are we going to do this, that, or the other? A good way to actually find those approaches is actually, so if you're studying lupus, go to the Lupus Foundation and look at what the new research is going on. Or if there's Dr. Riley in our department works on the aspects of complement and its role in autoimmune disease, and in particular lupus. You go talk to them. Just say, I'm really interested in what's going on. Where's the edge? You want to find the edge of the research. If you're interested in type 1 or type 2 diabetes, go to the Diabetes Association and look at what's the newest stuff. Or for that one, you can come talk to me because I'm a member of that association. Or cell biology, you go there and look at what's the newest thing about this particular protein that you want to do. But so you can interview people. And we're not limited to this campus. Go to the medical school. There's lots of people working on disease. They get paid because they work on a disease. That's why it's called biomedical science. Okay. So if you have questions about how to do this, then come talk to me. Groups that come talk to me usually do better because I can save them a lot of work by going, this idea is crap. This idea is really good. This idea might have some potential. Okay, and I can eliminate that because I've done this for a living for 35 years. Okay, so that's where you are with the group stuff. And okay, so just a real brief thing back to Back to the Future here. This is Bart Simpson's head, and I'm just showing you the diagram of why is it that protein, so the SGLT1 and GLUT5 are up and down lining this. There's so many proteins in this membrane, it's standing room only. You don't see that, but that's why you have Bart Simpson's little spiky hair, is to give more surface area so you can pack all the transport proteins in there. It's hard to do that. There's so many proteins. And what keeps them from diffusing through here to the basolateral membrane and going to the wrong spot is this tight junction. And there's actually a couple junctions. So this is not for you to memorize the anatomy of the intestine. It's just to show you that that little belt is in 3 dimensions all the way around the circumference of the cell. Okay. Now, I think what we're going to do is we're going to do a couple of questions. So get your clickers out. Try to do this one first. These should be pretty quick. These are sort of vocab type of questions. In fact, you should be able to do this in about 30 seconds. Open. At most. And everybody needs to click, so you're counted as present. If you didn't bring your clicker, or you forgot it, or fell asleep or something, you can sign in at the end of the class. So we're sort of ambivalent between answers 2 and 3. So if you didn't get your clicker in, we're going to do another problem, so don't worry about it. Pull and close. All right. Okay. Now get with your group very quickly and give me a group click on your answer. It looked like C and D, or was it B and C? I think C and D were the ones that people were coalescing around. Think about this function, not so much the structure. I got to open the puzzle. Actually, just one person should be clicking. We don't have 20 groups. We're growing groups as we look. Okay, I'm going to stop this and start it over again. Okay. Answer 3. Huh. Let's make sure this is 5. All right. Shoot it up here again. Oh, it's close. I'm sorry. Mine are falling asleep again. All right, that's fast. Okay, now let's hopefully. Okay. I don't know why it's not. So 95% said C, which is the correct answer. The reason it's correct is because that's an ion channel function. It's an ABC transporter, but it's not actually functioning as a transporter. So it is a poster child, so I'm not just going to give you a bizarre protein and make you decide what it is. You're supposed to know that one. And so everything else, that's a pump, and your book considers pumps transport. Circa, that's a pump, it's a transporter. Glut4 is a uniform, so it's a transporter, and that's a pump, and so it's a transporter. But that is functioning as a chloride channel. Okay. Let's do one more, I think. Closed. Okay. And let's clear this again. Okay, let's take about 30 more seconds. And just click if you don't get it. You can have no answer. You're going to need to beat the countdown. Okay, so answer 3 looks more popular. So that is C, 3 sodium dissociate, 2 Ks bind, and an aspartate residue is dephosphorylated. That is precisely right. So all 3 of those conditions have to happen. If this one's close, ATP is hydrolyzed, 3 sodiums dissociate, 2 Ks bind, but that won't make it flip. The 2 Ks binding cause the dephosphorylation, and then it flips. All right, so very specific on the transport cycle. All right. Yeah. Okay. Come back. Okay, so this is, we're going to move into ion channels is our last major protein, because everything's been a transporter up to this point. What makes ion channels so different? And they are, again, the target of a lot of drugs, so they're a lot of medical importance is attached to understanding their function. Okay, so we're going to understand the major differences between solute transport and ions versus everything else, categorize our ion channel examples into types, and then we're going to account for their properties. Like, why are they so specific? Why do things move so fast? Why are they selected? Why does voltage affect them? And then ultimately I'm going to give you a tool that you can use to determine which way an ion is going to move through the channel, because they can only go through there passively, so they must be going down electrochemical gradient. But it's not always simple, because sometimes the chemical potential for that ion opposes the electrical potential. If they're both pulling in the same direction, it's obvious which way it goes. The thing opens, it's going to move down. It's electrochemical gradient. But what if the electrical gradient is opposing, but the chemical potential is trying to push it? Which one's more powerful? Well, there's a way to figure that out. That's called the Nernst Equation. Okay, it's very straightforward. Okay, make sure how much time I have. Let's see. 10:35. Okay, so generally, let's start out with looking at the different kinds of channels. The solutes that are moved through those channels are typically inorganic small ions. So the solute is also important: potassium, sodium chloride, calcium. The energetics, as I've just gone over, is always passive. Okay? It's not simple diffusion, it's facilitated diffusion, because there's a protein involved called a channel. The kinetics is very, very fast. So slowest is a pump, 1 to 100, maybe on a good day up to 1,000 per second. Transporters, SGLT1, a little bit faster, anywhere from 100 to 10,000 per second. This is typically, that's 10 to the 8th per second. That's a lot. Almost a billion per second. So the kinetics are very fast, and this is the one case where the kinetics are not described by Michaelis-Menten kinetics. So we've put it in a separate category. We basically have two different kinds of channels, and I actually like to take you to this website. I'll give you the link because I really like it. It's University of Toronto, and they have a really nice way of talking about it. Let's see. Let's get the. I think this is. It's silent, but the picture says 1,000 words. Okay, so starting out, they categorize ion channels as specific and non-specific. Typically, ion channels are very specific. They're usually specific for one ion, right? So this would be I believe this is a potassium ion, so you click it. See, only the potassiums are moving through that, so that's a very specific channel. Most channels are like that, but we're going to talk about one. It's called the nicotinic cholinergic channel that is responsible for your skeletal muscle contraction. Is pretty low affinity. It's kind of sloppy. It will take calcium, sodium, and potassium. Why does it choose usually to have sodium? Well, we'll have to know more about the physiology before we can answer that question. So some are specific, some are non-specific. So we would call this a K channel, potassium channel. We would call that a cation channel because it is specific for cations over anions, but within the family of cations, positively charged ions, it's pretty sloppy. Okay, so that's one way of categorizing it. The other way of categorizing channels is whether or not they're open all the time or are they gated. That means they're open and closed depending on the circumstances. So if it's and there's lots of synonyms for this. If it's a passive ion channel, it's open all the time. We also call that a resting. So passive means resting channel, means it's always open, or it's also called a leak channel because ions are always leaking through the thing. And that plays actually a very important role in setting your membrane potential. Okay, so this is not just because they're passive doesn't mean they're unimportant. Okay? But most of the ones that we'll talk about are gated ion channels. There's something that opens and closes the gate, so the channel is closed until it's told to open, and it can be made to open based on a number of things. I think this first one that's a ligand that's actually binding to the channel and opening the gate, and as long as the gate's open, it's going to flow through there at a very fast rate, almost a billion per second, until the thing inactivates. Okay, so let's look at the different kinds of gated channels now that we know that they exist. So we have ligand gated. That's the one that we just saw there, and we'll look at a very specific example. That'll be the nicotinic cholinergic ion channel. It's gated by acetylcholine at your neuromuscular junction. That's why all of you can move at the moment. It's because that thing's working. All right. So ligand gated is where a ligand tells the thing to open. And by the way, your book seems to distinguish between extracellular ligands like neurotransmitters and intracellular ligands. I do not. Both of those are called ligand gated channels. You've actually seen an example of an internal intracellular ligand gated channel. What's it called? It was the answer to one of the questions. CFTR. That's gated by ATP. ATP has to bind to open the gate. Okay. Okay. So now this one is a little funny. Okay. Raises its arm. So this one is actually going to open when the membrane potential depolarizes. So what does that mean? It means the inside is normally minus 60, minus 70. So that means this becomes less positive. So when it's in the resting state, as long as you have, this is like minus 60 or 60 millivolts more positive outside than it is inside. And so it's got little positive little alpha helices that are being repelled by that positive charge. But when this cell depolarizes, the membrane potential becomes less positive outside, and what that does is, if this is becoming more negative, it pulls on these little arms, and that's what opens the channel. So let's click on it one more time. See, it became more negative, and that attracted the sodiums. The channel opened, and the ions moved through there. Our example will be a potassium-gated or potassium membrane potential gated channel, voltage gated channel that moves potassium. But we won't get to it in detail today because we're going to just go through the introduction. All right, so here we go. So the third class is mechanically gated. So something deforms the plasma membrane and it senses that mechanical deformation and then opens. A good example of that is a channel in your ear, your inner ear. In your cochlea, there are cells that are called hair cells. And sound waves are transferred into the cochlea as pressure, and that fluid pressure deforms the membrane. It activates, it opens the ion channel in the hair cell, and you send a nerve impulse to your brain that's interpreted as music or sound. Okay? So that's how sound is deconvoluted in your brain. Translate those nerve impulses into something that is sound. Okay? It could be music, it could be anything. But pressure is something that can gate a channel. All right, so the last one, and this one is not in your textbook, but I did put it on the outline, phosphorylated channel. So you can gate a channel by changing the phosphorylation state. And guess what? You have a channel already that does that. It is, you want to talk about what channel? CFTR. It's both ligand gated and it's phosphorylated, because you have to phosphorylate it before you can put the ligand on it. So it's both phosphorylated and ligand gated. So it's a good example of both of those classes. So we go like this, stick a phosphate on there, opens the gate, things flow through, then it closes. Okay. All right. Enough about that. So let's close this for a moment. So your book shows a couple of these, but not all of them. Ligand gated, mechanically gated, voltage gated, but then the one that's missing is phosphorylation as a gating mechanism. Okay, here's the deal. Again, with ion channels, this is too simple. Okay, you cannot use, it's sort of like the allosteric enzymes versus the classical Michaelis-Menten enzymes. Allosteric enzyme, you cannot use that equation. You cannot use the Michaelis-Menten equation to characterize the rate of movement of ions as a function of ion concentration. In fact, it depends on the membrane potential, and you can just plug it into this equation. Take a moment to commit that to memory. It's quite nonlinear. We're not going to deal with it, right? So it's a complicated nonlinear equation between current flow and membrane potential, right? All right, so fortunately for you, we're not going to do this. So the last thing that we'll talk about today is the most simplest ion channel that we can talk about, and actually Dr. Coelho over at the medical school yesterday does his work on this bacterial channel. This is a bacterial potassium channel. It's so simple, it just has actually 2 alpha helices, okay? But you can't make an ion channel pore out of 2 alpha helices, so there has to be 4 different subunits, each having what's called an S5 and an S6 helix. So each subunit has 2 alpha helices, but it takes 8 of these alpha helices to create this pore, 2 from each of the 4 subunits. Now, what we've done is we've removed the one that would be in this side, and we've moved the one in the back. So we're just looking at 2 of the subunits so we get a good view of what's going on. So this is inside the bacterial cell. And what's happening is there's a potassium ion will go into this pore. Not by itself, the potassium ion will be coordinated by 4 different water molecules. So potassium doesn't go around by itself, it's hydrated. Okay? It's got we know exactly how many water molecules it has. It's big and bulky now because it's holding on to 4 water molecules, and it can sneak through this little hole right here. Why is it going in? It's because these alpha helices have little loops on them that have negative residues. Aspartic acid, glutamic acid. So these negative residues in the sequence right here is beckoning the potassium ion, which is positively charged, to come in the door. Okay? So that's what's recruiting. That makes this cation selective, is those negative charges. So positively charged ions like negatively charged anions like chloride would be repelled because of this little gate right here. It's not a gate, but it's the first level that you have to pass through. So it pops through here into this area that's sort of big. It's called the vestibule. And it actually penetrates almost halfway through the lipid bilayer before it reaches this little special spot. Okay? What's sucking it in here is these 2 pore helices. So let's look at the anatomy of this. So the outer helix is S5. It breaks up into a loop, forms a small pore helix, and then it forms what's called the selectivity loop. Okay? This is actually going to line the pore. Okay? But the pore helix has a partial negative charge that's aimed right at the entryway into the selectivity filter. So it's causing the potassium ion, instead of to float around in here, it's being sucked into the selectivity filter. Okay? So the problem is it can't get in there with 4 water molecules. That's very stable, okay, the 4 water molecules, but it can't fit in there. So what happens is this selectivity loop dehydrates it. It strips off the 4 water molecules, and then the dehydrated potassium can then jump into this, what's called a selectivity loop. You can see the motif is just a loop, okay? And it's called the selectivity loop because this specifies potassium. Nothing can go through this except at any appreciable rate except potassium. Okay? And what's selecting for potassium is not the R groups. So this is glycine, tyrosine, glycine. It's a repeated sequence on each of the 4 different subunits on that loop. But it's not the R group that is creating the selectivity filter. It's the carbonyl group. That's why it shows these carbonyls projecting into the pore. Carbonyl has a partial negative charge, so it's going to really like a naked, dehydrated potassium ion. Why is that selecting for potassium? This is why. So water, the potassium is very stable as long as it's hydrated to 4 water molecules. So when you dehydrate it, you better come up with some way of coordinating that ion that's equally low free energy state. And the carbonyls exactly replicate the position of those oxygens in the water. So the water is allowed to go away, and then it's still perfectly coordinated inside the selectivity loop. Look what happens when you try to put sodium in there. Sodium likes 4 water molecules, right? But it doesn't fit into the selectivity loop. It's the way it's arranged is it can only contact 2 out of the 4 different carbonyl groups. So sodium is not allowed to go through. So the specificity is determined by the selectivity loop. And what holds it in there is the S5 and S6 segment is positioning it at the appropriate position. So this loop is actually located between S5 and S6. Okay? So everything has a function in this. And I think we're just about out of time. Yes. We'll go back and use this information next time.
Yeah. All right. Your second was right here. I took her off. She had to leave her.
Okay.
And this is her. So
Okay.
is that okay?
That's okay.
Do I just tell somebody or?
Well, it's okay. She left early. She probably, she clicked in.
No,
Well, you just put the name down.
Okay, so the dephosphorylation of the
Aspartate.
yeah, I thought the deassociation of the potassium is what causes it to go back to the E1 conformation. That's what I was confused about.
Right, but it's
But
no. That's the ATP binding causes the 2 potassiums to dissociate.
Right.
But it's already in the E1.
Ah.
Yeah, it just completely returns it to the original state,
Okay,
which is
right.
empty, ready with ATP bound.
Okay, but once that phosphate group is clipped off, that's when it's back
It
to the
refolds to E1.
Okay. All right.
It is confusing. Well, the problem is, yeah, you can do that. What's the issue? Because it's never been done. I mean, you're welcome. You'll have it.